Good morning and welcome. My name is Barbara Scott. I'm a drug substance CMC quality assessor in the Office of Pharmaceutical Quality, Division of Lifecycle API. Today I'd like to share with you some of the key concepts surrounding the agency's expectations for conducting and submitting a hazard assessment in order to classify impurities based on the ICH-M7 guideline. Our goal is to give industry a clear understanding of what the agency's expectations are for a complete impurity hazard assessment so that you can provide us with all of the information we need in order to evaluate your submission from both a quality and safety perspective. During today's talk, I'll give you a brief background and discuss the scope and key concepts of ICH-M7. We will review the hazard assessment steps and how to use the hazard assessment to classify impurities. I'll take you through an example hazard assessment and how the information in it is used by the agency to evaluate impurity control. Because the control of any impurity is dependent on the intended drug product, we will look at a sample calculation for acceptable limits, taking into consideration the intended drug product indication, treatment duration, and maximum daily dose per the labeling. Last, I'll provide some considerations on what to submit for analytical methods used to support the options selected for control of mutagenic impurities. There are 3 documents dealing with ICH-M7 that I want to draw your awareness to: the main guideline, the addendum, and the most recent work of the ICH Expert Working Group, the ICH-M7 Q&A document. The main guideline, Assessment and Control of DNA Reactive Impurities in Pharmaceuticals to Limit Potential Carcinogenic Risk, reached step 4 6 years ago in June of 2014. The addendum is a list of 14 Class 1 common pharmaceutical impurities with compound-specific acceptable intakes, or PDEs. The addendum reached step 4 in March of 2017 and is currently being updated by the ICH Expert Working Group to include additional impurities. Last, the ICH-M7 Q&A document is a supporting document which provides clarification on both safety and quality aspects of the main guideline. This document reached ICH Step 2B in June of 2020 and went out for public comment. The commenting period has now closed, and the ICH-M7 Working Group is currently addressing stakeholder comments. There are several ICH guidelines that apply to impurities. Classification of impurities requires application of the correct guideline. This slide reviews the scope of ICH-M7, which is discussed in detail in Section 2 of the main guideline, as well as in the 3 Q&As listed here. Bulleted here are the drug substances and drug products that are out of scope. Biological, biotechnological, peptides, oligonucleotides, radiopharmaceutical, fermentation, herbal, crude products of animal or plant origin are all out of scope. Drugs intended to be used for advanced cancer that are defined by ICH-M7 and ICH-S9 are out of scope. APIs that are genotoxic at therapeutic concentrations are also out of scope. Exposure to mutagenic impurities in these cases wouldn't significantly add to the cancer risk of the drug substance, and therefore impurities found in those APIs can be controlled at acceptable levels for non-mutagenic impurities. And last, already approved drug products are out of scope unless one of these scenarios exists. There are changes to the drug substance manufacturing that results in new impurities or increased acceptance criteria for existing impurities. Or there are changes to the drug product formulation or process that result in new degradants or increased acceptance criteria for existing degradants. Or there is a change in the drug product indication or dosing regimen that would significantly impact acceptable cancer risk. In each of these cases, ICH-M7 would apply, and each submission would be reviewed on a case-by-case within the agency. CMC quality and safety groups at the agency collaborate to evaluate the actual and potential impurities that could be present in both drug substances and drug products. Quality reviewers look at the route of synthesis of the drug substance and identify the process impurities that could carry over into the drug substance. They also assess the stability of the drug substance to determine what degradants are likely to form and whether or not they are present in the drug product. Sources of these impurities are shown on the left and could result from carryover of starting materials, synthetic intermediates, or reasonably expected byproducts, for example, oxidation reactions. For each impurity and in close consultation with our safety colleagues, the quality reviewer evaluates industry's hazard assessment, impurity classification, control, and the analytical method for acceptability. The next few slides look in detail how the M7 guideline defines the impurity hazard assessment, classification, and proposed control. The hazard assessment. What is it? Per Section 6, the hazard assessment involves an initial analysis of actual and potential impurities by conducting database and literature searches for carcinogenicity and bacterial mutagenicity data in order to classify them as Class 1, 2, or 5 according to Table 1. If data for such a classification are not available, an assessment of structure-activity relationships that focuses on bacterial mutagenicity predictions should be performed. This is known as QSAR. This could lead to a classification of the impurity into Class 3, 4, or 5. The translation here is that if literature isn't found or an AIM study performed for a given impurity, then ICH M7 compliant QSAR model should be run in order to bucket the impurity. You'll hear more from my colleague, Dr. Krulak, in the next presentation on the agency's expectations for the application of QSAR and expert knowledge to classify impurities. It's critical to note that visual inspection of a compound for a structural alert in order to classify it according to Table 1 in the guideline is not M7 compliance. So let's look at Table 1 in more detail. The impurity classifications are defined, and the resulting control actions are outlined here and in Section 6 of ICH M7. There are 5 classes or buckets. 1 through 3 are mutagenic, and 4 through 5 are non-mutagenic. In the initial hazard assessment, industry should search known literature. If data is located, it can be referenced in the hazard assessment to support classification of an impurity. Depending on the literature study outcome, the impurity may be Class 1, known mutagenic carcinogen, or Class 2, known mutagen but with unknown cancer risk, or Class 5, a non-mutagenic impurity. When no literature data is found for the structure, QSAR is run to screen for structural alerts. The guidance says that if M7 compliant models are run and the QSAR prediction is negative, the impurity can be classified as Class 5. If the QSAR prediction is positive, then the impurity is Class 3. In cases where a valid AIM study is performed as a follow-up study, a Class 3 impurity can be downgraded to Class 5. An impurity can be assigned to Class 4 when the QSAR prediction is positive, but the same alert is shared with the drug substance which tested negative in the bacterial mutagenicity assay. In this case, by structural similarity arguments, the impurity would be considered negative and non-mutagenic. Once the impurities are appropriately classified, Table 1 outlines the proposed action for control as shown in the last column. Classes 1 through 3 need to be controlled at acceptable intakes based on ICH M7, and classes 4 through 5 can be controlled per ICH Q3A or B. We've looked at the hazard assessment elements, defined classes of impurities, and discussed the acceptable intakes. On this slide, we provide an example of what the agency would like to see from you in a hazard assessment summary table when you submit your DMF or drug substance to the agency for review. The table should include the structure, the chemical name. The hazard assessment column should include links to literature studies, the overall QSAR output from the required expert rule-based and statistical-based models, and then based on the hazard assessment, the proposed impurity classification and the corresponding action for control should be indicated. Once the hazard assessment has been provided and the impurity classification proposed, the quality and safety assessors work together to review the data provided. For literature justifications or AIM studies performed by the DMF holder or applicant, our farm tox assessors ensure that the literature supports the classification, and if AIM studies were performed, that they are fit for purpose and appropriate based on the intended drug product. All QSAR predictions and any expert review provided by a DMF holder or applicant is consulted to our computational toxicology consultation service to verify or override submitted predictions, as well as to provide feedback, if applicable, on the acceptability of the models that might have been used. For safety verified class 1, 2, or 3 impurities, the quality chemist then assesses the holder or applicant's proposed monitoring, limit, and analytical method. For mutagenic impurities, the monitoring options are outlined in sections 8 of the main guideline. They're designed to give industry a large amount of flexibility in where controls are implemented based on risk. For class 1, 2, and 3 impurities, quality control options 1 through 4 are available. To review, options 1 and 2 require a test at drug substance release or upstream at TTC. Option 3 allows for an upstream test higher than TCC with an appropriate spike purge study, which demonstrates the impurity is purged to 30% TTC in the drug substance. Option 4 allows for no monitoring based on a robust process design and understanding of the purge pathway for a given impurity. For option 4, purging of the impurity to negligible levels or 1% TTC is recommended. For class 3 compounds, which are impurities with a confirmed positive QSAR prediction, there's an additional justification pathway that is outlined by the guidance. As an alternative to controlling the impurity by options 1 through 4 that we just discussed, you have an option to choose or to perform a bacterial mutagenicity study, which is typically an AIMS test, on the impurity and submit it to the agency for review. In cases where the agency determines the study is valid, these results would overrule any structure-based concern or QSAR prediction. On this slide, we'll pull together all the information we've discussed on the previous slides and consider a hypothetical example hazard assessment submitted to the agency for drug substance APIX. In this example, the table contains 4 impurities with structures, names, hazard assessment, classification, and an action for control. Let's look at impurities 2 and 4 in more detail. For impurity 2, the DMF holder or applicant has not submitted any literature data, but did run the required orthogonal QSAR methods to screen for a structural alert. In this case, both models fired an aromatic nitro group as an alerting structure, and the firm has placed this impurity into the class 3 bucket. The firm then proposes to control this impurity in the drug substance release specification at TTC rather than perform a follow-up AIMS test. From both a quality and safety perspective, this would be an acceptable control option per M7 option 1. Looking at impurity 4, the firm has provided literature study, including a positive AIMS result and CARSI study. They didn't perform QSAR and classified the impurity as a class 1 mutagen. The DMF holder or applicant then proposed an acceptable intake of 157 micrograms per day based on the literature with control upstream at that level. As I said earlier, since this is consultable information, the safety assessment would fall on our farm tox group. If they found the acceptable intake justified based on the literature provided, then from a quality perspective, the proposed option 2 action for control would be acceptable. For option 1 and 2 control action, the proposed impurity limit in the drug substance specification or upstream control needs to be calculated as shown by the equation here. For an individual impurity, the limit is equal to the acceptable intake in micrograms per day divided by the maximum daily dose in grams per day of the intended drug product. Both the acceptable intake and maximum daily dose are variables here. On the next slide, we'll review where to find this information. To determine the acceptable intake and maximum daily dose, there are a few locations to look first. Is the impurity listed in the ICH M7 addendum? If so, use the acceptable intake listed there. Otherwise, look at the labeling of the intended drug product to determine the duration of use. For example, is the drug product designed to be taken daily for greater than 10 years, or does it have a duration of use less than 1 month? After the duration of use is determined, Table 2 in M7 gives the acceptable intakes allowed for each individual impurity. Another consideration to acceptable intake requires examination of the chemical structure of the impurity. If it's a monofunctional alkyl chloride, it's eligible for a 10-fold allowable increase in the default acceptable intake. And I refer you to Note 5 in ICH M7. Likewise, to determine the maximum daily dose, the reference listed drug label should be consulted. If more than one indication is present with different daily doses, please use the maximum reference daily dose when you're calculating the limits and developing analytical methods. In this slide, we'll go back now to our earlier hypothetical example for APIX and Impurity 2, which was proposed to be monitored at the drug substance release per option 1. We'll walk through the steps to find the limit that should be proposed in the release spec and used for analytical method development. So let's say APIX has been determined to be in scope of ICH M7 and the intended drug product used for greater than 10 years to lifetime. Per Table 2, acceptable intake is 1.5 micrograms per day. Per the reference listed drug label, the maximum daily dose was noted to be 150 milligrams per day. Doing the math, the limit for the impurity is calculated to be not more than 10 parts per million. Recall that in the hypothetical example provided, the firm proposed option 1 control for Impurity 2. The CMC reviewer would look for the test and calculated limit in the release specification. They would review the corresponding analytical test method and all validation work to ensure that the method was suitable for use. An important note here is that if your APIX is repurposed for use in a different drug product or in combination with another drug, where the acceptable intake, indication, or MDD changed, the limit as well as the analytical method used could be impacted. So please keep this in mind. This brings me to the last aspect I want to talk about today, analytical methods used for impurities with mutagenic risk. For the analysis of mutagenic impurities, the analytical methods need to be both specific and sensitive. Limits of quantitation are typically in the range of 0.3 to 5 parts per million, and oftentimes use HPLC or GC coupled to mass spec. The limit of quantitation, or LOQ, should be set at the acceptable limit, but preferably at 30% of API. And I'll refer you to Section 8, Subsection A, Options 1 and 3 of the main guideline for more information. Full validation of release methods are required for Option 1 control. In cases where other options are chosen or spike purge studies are performed, due to the sensitivity of the requirements, it's recommended that the method description, system suitability, specificity, linearity and range, accuracy, precision, and LOD/LOQ be provided. This is for ease of agency review. In summary, we have gone over the elements contained in a hazard assessment and what the agency's expectations are for classification and actions for control. We have clarified that classification of impurity by visual inspection for structural alerts is not M7 compliance, and the expectation is that QSAR will be run. We have described how the agency reviews the hazard assessment to ensure patient safety through the appropriate impurity classification and monitoring during the process with acceptable limits and analytical methods. I hope you found this presentation helpful and that you can use the information here to assist in providing the agency a complete impurity hazard assessment in your future submissions. After this presentation, if you have any questions, please type them into the Q&A box in the bottom right-hand corner of your screen so that we can address them during the scheduled Q&A panel after this session. If you have a question on this presentation after the workshop's over, send them to the email address provided here by March 19th for inclusion in the follow-on webinar April 9th. For additional information on this topic, please refer to the presentations given today by my colleagues, Hongbao Lai Naomi Krulak, Chantal Gupta regarding impurity qualification, and the poster by David Green, which highlights several questions from the ICH M7 Q&A supporting document. This leads me to the end of my presentation. I'd like to thank you for your kind attention, and I look forward to the Q&A panel. I would also like to acknowledge and thank the wonderful people on this slide for their help in the preparation of this talk and for their technical expertise.